Dr. Sharon Savage is going to be our next speaker. Dr. Sharon Savage is the chief of the Clinical Genetics Branch in the Division of Cancer of Epidemiology and Genetics at the NCI. Okay. Her impactful research work has led to improvements in the diagnosis of TBDs and significant advancements in the understanding of TBDs. She will be presenting the popular TBD 101 session for us today. From those of you that have been in the community for years and are familiar with TBDs to those of you who are just recently joining the space. TBD 101 is an incredible introduction into the world of telomere biology disorder. It lays the foundation for the what and whys of TBDs. So thank you for all, and welcome Dr. Savage.
Thank you. So first of all, welcome everyone. It's great to see so many familiar faces, and as much as I really love seeing you, I'm also super excited to see new faces to the community. As Katie said, the group really had very little time to kind of put this together, so I think it's going to be a great program, and I'm really happy to be here and with my team, who you'll meet over the course of the day as well. So just to get started, I have a lot of information here, but I did borrow a slide from Dr. Alter, who many of you know, Blanche Alter, who reminds us, and this is something we hear in the medical community, we're taught if you hear hoofbeats, chances are it's a horse, right? Because that's much more common than, say, a zebra. But zebras also have hoofbeats, and so in the medical world, a zebra is an unusual or rare event. But what we're talking about here are really those unicorns, right? The ultra rare features, and this is where in the medical world, we're not always trained to recognize unicorns because common things are common, right? And this is the challenge that everyone here faces. I pulled this quote from our needs assessment study that the team back there, Kat, Cam, and Emily have been integral in. And this was a quote from a study participant who says, "What is actually having short telomeres? I mean, what does that actually mean? When do things start falling or when failing? Or when do I start, if I can't generate new cells, what should I be aware of? But what does that mean in the timeline of your life?" And I've been sharing this with scientific talks as well because I think it really helps put a framework on a lot of what we think about with telomeres and TBDs. So what are telomeres? This is kind of going to go over a whole bunch of different things. So telomeres are at the ends of our chromosomes, and that's where our DNA is packaged. Ashley is going to talk about that in the genetic counseling session in a little bit more detail. But basically, telomeres were discovered by Barbara McClintock in the 1950s when she exposed corn or maize DNA chromosomes to radiation and found they actually the chromosome ends stuck together. That was kind of the first discovery that, there must be something protecting the ends of chromosomes. And then the term telomere came about from Hermann Mueller, who combined two Greek words meaning end and part. So we got telomeres. So telomeres protect the ends of chromosomes for basically anything that has any organism that has a nucleus. So algae, there's a lot of work actually in the telomeres biology world in studying single-cell organisms, and everywhere all the way up to humans and everything in between. We all have telomeres. These are those little yellow dots at the ends of chromosomes in this image. But telomeres are much more than just DNA at chromosome ends. They're protected by this complex network of proteins that serve to help cap the telomere, help protect those ends of the DNA, but also help to open it up and allow it to lengthen. And to basically, it's a balancing act of those telomere proteins. So, but why the telomeres, why do those ends of our chromosome, the ends of our DNA need protecting? It's because DNA damage is a constant threat to all of us. It's a matter of life. So telomeres and DNA is sensitive to things like ultraviolet radiation, changes in our metabolism, chemical exposures, and even cell division, cellular replication, and growth that we all have over time is also a stress on our telomeres. So the problem is that if telomeres don't work, if they're not well protected, bad things can happen. This is a very extreme example. This is a study looking at mouse chromosomes, And in this example on the right, you can see that these are telomeres missing a critical part of that protein complex I just showed, and you can see the chromosomes stick together. And that's bad because you're going to get more DNA damage in this kind of constant cycle, and it's not compatible with healthy cell division or cell life. So telomeres also get shorter as we age. It's a normal part of aging. And that's because of what we call the end replication problem. So every time a cell divides, the cell has to replicate its entire DNA content. Every single chromosome has to replicate itself. And the problem is at the ends of those chromosomes, the DNA replication machinery just doesn't work that great in getting that ending completely extended. So what happens over time is that the analogy that's often used is the aglet, which is this plastic cap at the end of your shoelace. When you have a brand new pair of shoes, brand new shoelaces, right, that cap looks perfect, it's pristine. And then over time, the plastic end kind of wears away, starts fraying, and eventually your shoelace gets shorter because it just frays down if you don't fix that. So that is kind of what happens to telomeres over age. Normal phenomenon happens to all of us. We also know that we have measures of how telomeres change over time in the general population. So these are graphs that are actually based on telomere-like testing for patients with TBDs. But what you can see is from 0 to 100, based on unaffected general population controls, you see this decline in those colored lines in telomere length. What I'll also, though, just emphasize is if you look at these colored lines, this is the normal decline in telomere length with age. You see a bit of a steeper curve in early childhood. That's simply because kids are growing like crazy, so their cells are dividing because you gotta go from a 7-pound infant to, if you're like a 200-pound, 6'5" teenager, which I may or don't have. And so you can imagine the number of cells that have to divide in a short time, right, to make that huge growth. So you see this change over time, and that's why anytime you do telomere length measurements, it's really important to compare with normal age-adjusted controls. But what I also want to point out here is there's this wide range. So everyone who is used to kind of going to the pediatrician with young children, you see very quickly about weight and height percentiles, right? And you want to know, where does your kid fall? Are they on the shorter side, on the taller side, heavier, lighter, etc.? And you follow their growth curves. These are essentially those same kind of curves, and what you see is a lot of variability. The first percentile is the bottom, and the blue is the 99th percentile. So that means 99% of people in this population have telomere lengths that fall in that range, and there you can see huge variability in populations. So you would see, well, why don't we want our telomeres to just get longer? Why not just extend, just whatever we can, make telomeres longer, and that would be great, right? The thing is that telomeres also are very much a protective mechanism because they basically limit the number of times a cell can divide, and that is important because it reduces the possibility of that cell getting DNA damage. So over time, with number of cell divisions, telomeres get shorter, and eventually cells will either undergo what we call apoptosis, which is cell death, or they just kind of become these quiet senescent cells that just kind of sit there and don't do a lot except cause a little bit of trouble related to inflammation. And this is important because telomeres have two different roles, the way I think about it, in cancer development. So again, with each cell division, telomeres getting shorter and shorter. If telomeres get to a critically short length and the cell does not die, what can happen is those chromosomes can stick together again, or they'll abnormally upregulate some other proteins, and then that will keep those cells dividing when they really shouldn't have been. In cancer as well, what we see is often telomeres are lengthened because cells kind of trick their DNA and they do some things to start making telomeres longer and longer. And the problem there is that you have cells that are able to continue to divide and maybe they shouldn't be because they have reached an endpoint where they have enough DNA damage that they shouldn't have be able to divide. So there's sort of two different roles as you start thinking about uncontrolled cell division, which is where we think of with cancer. So telomere biology and in TBDs and in the general population, I really think of it as a very complex balancing act. So now let's talk a little bit about TBDs. And I put this long list of names because, as Katie mentioned, we started out as dyskeratosis congenita outreach with this group, and lots of different names that we will touch on, but they're really all connected by biology, and that's what I'm going to talk about next. So I now consider a telomere biology disorder, and I think this is sort of where the field is at, is it's a spectrum of often, but not always, inherited disorders caused by rare DNA changes in genes that are essential for telomere biology. So genes that are essential for all those things I just kind of briefly described about telomere biology. But how did we get here? So the first studies, actually the first report of what we now call dyskeratosis congenita, or DC, was actually around 1906. And there were early reports in the early 20th century looking at that first described the skin pigmentation changes, oral leukoplakia, abnormal nails, eventually found that they also had abnormal blood counts. And these were first described by dermatologists, and that's how it got the name DIS. It just means abnormal. Keratin or keratosis, keratin is a key part of our skin and hair and nails. That's the protein part. And congenita just means that you're born with it. So this is one of those very classic, typical early DC families that were described, where what we have here, and again, Ashley's going to talk about this a little bit more, is excellent inheritance, meaning that only boys are affected, the moms are healthy, and have one abnormal X chromosome that gets passed on to boys only. And this is the classic nails, skin, white spots in the mouth, that oral leukoplakia, and the inheritance pattern. So at first, when I started working on these diseases about 20 years ago, the majority were thought to be X-linked. We thought we had the answer. And that's because we had also discovered, the field had, Inderjeet Dogal's group in the UK, had found that germline mutations in a gene called dyskerin was responsible for dyskeratosis congenita, and that mutation caused very short telomeres in the patient's cells. And that was the very first time that telomere biology was connected with human disease in this way, in an inherited mode. And it accounted for around 20% of the cases or so at the time. So that was around 1999 to 2004. And when we go back to the literature and to what we were seeing around 2000, was actually the first report of a female case was in 1963. And subsequently, around 2004 and 5, there were discoveries that found there were mutations in telomerase, in TERK, its RNA component. Those are the proteins that extend the telomere lengths. And this was the first time this added to the fact that we had autism. We had dominant or recessive inheritance. And again, Ashley's going to go into those in more detail. But basically, we expanded now. In 2005, we had 3 genes, and we had different modes of inheritance. So this allowed us, working with my group, with Dr. Blanche Alter and Dr. Peter Lansdorp, who's at the University of British Columbia, developed the method that we now know is the standard diagnostic test for TBDs, which is measuring telomere lengths in white blood cells by something we call flow FISH. And it requires fresh white blood cells. They get run through a machine, and you stain the DNA and the telomere ends, and you can quantify how long those telomeres are. And what we showed quite a while ago now was that people with DC, who are in these colored symbols down here, or TBDs, clinically had very, very short telomeres for their age. And this really helped us not only diagnose the disease, but also was the foundation for what has now become, over the last decade plus, really 2 decades almost, of gene discoveries as well. Because in addition to clinical features, we now had a biologic marker that we could use to help us discover new genes. So I'm going to go through a whole bunch of different TBD complications. This is, again, meant to be an overview, and a lot of these you're going to hear about in much more detail over the next 2 days. And I just really want to emphasize that people with TBDs may face these problems, they may not. Everyone is very different, and the field has really just grown so much over the last 15 years that things are changing every day. And I love this quote from Dr. Seuss, because today, and I add it, and always, you are you, that is truer than true. There is no one alive who is youer than you. So even though I'm going to go through a whole bunch of different stuff, know that, again, it all comes down to you or your child or your loved one. So these are the classic diagnostic triad, we call it. So this is the features of dysplastic nails, abnormal fingernails and toenails. The skin pigmentation abnormalities can look quite different. On one hand, it's sort of this lacy, reddish, kind of reticulated pigmentation here. You see darker and lighter areas of the neck is where it's classically defined, but we know it occurs all over the body and is worse with sun exposure. Oral leukoplakia are those white spots in the mouth that often look like some of these, but they're not always white. Sometimes they can be red, sometimes they can be brownish, and they're something that we follow closely. A lot of folks are dealing with complications related to their blood system, so we're going to have a talk about bone marrow transplant tomorrow or the next day. So there's bone marrow failure, which is the failure to produce enough blood cells for your body. Patients with TBDs are also at risk of something we call myelodysplastic syndrome, which means that those blood cells, they try their best, but they're not quite normal, and that can potentially be a precursor to a leukemia. So that's why we watch the bone marrow very carefully, and bone marrow transplant is a good therapeutic option. In the respiratory system, our lungs, we know people are at risk of pulmonary fibrosis, head and neck squamous cell cancers, predominantly in the oral cavity. Our risk, pulmonary arteriovenous malformations are a newer kind of feature that, as we've studied more patients, we've seen this. And these are very small, abnormal connections between the arteries and the veins in our lungs or elsewhere that are very difficult to treat and can cause problems with oxygenation. You can also have those kind of problems in your gastrointestinal tract. Sometimes patients with TBDs have liver abnormalities, and that can be very complicated. Some young patients have poor absorption of food and nutrition. Esophageal stenosis, so narrowing of the esophagus, is often a problem. And anogenital cancers are also increased. Additionally, men often have problems with narrowing of the urethra that sometimes needs dilation and can be challenging to manage. Patients who present often early in life sometimes have problems with their central nervous system, including a small cerebellum, and that's this part in the sort of the back of your brain that's important, that's responsible for balance and speech coordination. Some patients have small heads, developmental delays, a wide array of issues, and some psychiatric illnesses, including anxiety, depression, and some other things have been reported. The skeletal system, again, we're learning more about this as well, in that we see that people sometimes have what we call avascular necrosis of the shoulders or hips, and requiring joint replacements. We've seen patients who have fractures of bones for minimal trauma, and that's something that we're still trying to study and understand. Eye-related complications include lacrimal duct stenosis, so that's your tear duct, can get narrow, just like the urethra can, or the esophagus. That can cause challenges. Abnormal eyelash growth, and then abnormal blood vessels in the retina in the back of your eye. So I just put down a big list of a whole bunch of complications that, again, coming back to connecting to telomere biology, how are they all even connected? And that's, again, because of these rare changes or rare variants, rare mutations, there's lots of different words used now that are usually inherited, but sometimes they happen what we call de novo, and you're the first person in the family with this genetic change. So when I first came to camp in 2010, I think we had 4 or 5 genes, 5 I think, And now we're up to at least 18 different genes that if they have an abnormality, they can cause a TBD. A challenge in the field is because now we have so many genes, most of these genes though are still very rare. So there are more common ones, but it's a challenge. Sometimes we have very rare genes. We have some families who have their very own gene, and it's hard to counsel them and elsewhere because sometimes we just don't know exactly how that's going to proceed. So I'm just going to put some of those clinical features in context with the way I think about this. Now I'm going to go through quickly because I'm running out of time. The way I think about the telomere biology disorders is I kind of group them in this way. These are the severe, young, everyone's severe. These are the early onset kids who are affected often even as infants or toddlers. Classic DC, Hoyer-Alder-Peterson syndrome, Coates plus, Revenge syndrome. These are all related and similar in that their telomeres are really, really short for their age, okay? And we think that's part of why they're so sick so early. And they are more likely to have autosomal recessive, meaning they have two abnormal copies of a gene, of a mutation in their gene, X-linked, that discarin gene, or a new TNF2 mutation in their family. Kind of at the next end, we have people who might present as adults with only one feature at the beginning. So they might have aplastic anemia or bone marrow failure, myelodysplastic syndrome, which I mentioned. We've had some patients who developed a head and neck squamous cell carcinoma in their 30s, which is a really early age for that cancer, usually as people over 60. Pulmonary fibrosis we now recognize as a very common aspect of the TBDs, and then liver disease as well. These patients can sometimes be really tricky because their telomeres are kind of in the 1st to 10th percentile. Now technically, telomeres in the 1st to 10th percentile are normal because 10% of people are going to have telomeres there. So we combine that with the clinical features and the telomere length and finding a genetic cause, and of which those are more likely to be autosomal dominant. That means these are patients who have one abnormal copy of a gene and one normal. So just briefly, I'm going to touch on a new study that we published last year looking at disease progression and clinical outcomes in the TBDs. Marina Nivich, who was already mentioned, led this study in collaboration with our team, and I'll just point out Dr. Alter, who retired 2 years ago, who's been at camp from the beginning, Dr. Neelam Giri, who now leads all the clinical aspects of our study. Some of you may have spoken with Stephanie Steinbart here in the middle, our referral nurse, or with Ann Carr, our genetic counselor, and others. So this is our team. And what Marina looked at was in our study at the NCI, we have 221 patients from 124 families. About just over half we had seen at the NIH Clinical Center. Some of you have been there, and we're grateful for that, to evaluate not just the genetics, but also the clinical features of the disease. And Marina looked at first how the males and females were similar or different. And what this shows is that we look just at overall survival. This is a 50% overall survival probability of around 55 years of age. And we see that males here look like they don't do quite as well as the females, and that's because the discarin, the DPC1 patients tend to have that earlier onset disease. Marina also took the chance to start understanding how the different genetic background is associated with clinical features and outcomes. And kind of similar to what we saw in the clinic, we saw that patients who have recessive disease, TNF2 disease, are likely to have poorer outcomes long-term than those who have the heterozygous autosomal dominant disease. And I just put this up here because I just want to say I don't want you to focus on the numbers. This is where we need to focus, is on working together to figure out how we're going to make those curves look better next time. Putting numbers on it as a scientist and as a doctor really helps, because then we can say, okay, this is an important aspect of biology. This is what we need to know. This is where we need to study it more. And things have changed a lot. I can tell you, if I had those same data from 10 years ago, we wouldn't be where we are now. And I really think we're making major inroads in doing this. Bone marrow transplant for patients with TBDs is going so much better than it used to even 10 years ago, right? Lung transplants are going well. We want to get away from organ transplants. I don't want to give anyone an organ transplant if we don't have to, right? So now there's a lot of new biology that we're understanding that are giving us important clues as to how to start treating this and preventing complications. So I want to end on that and emphasize that the guidelines book is an important starting point. I send it, anytime I get a physician who contacts me about the disease, I send them to the book as well. And I hope that everyone has had an opportunity to take a look at it. And I actually have the same photos that Katie had of camp. And I'll be around for the next 2-3 days. So feel free to come find me with any questions. Thanks.